A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Chief Operating Officer of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members and the broader scientific community about topics relating to antibody discovery and development. Our expert speaker, Dr. Francisco Ylera, will discuss the versatility of modular antibodies, and in particular, the use of SpyTag, SpyCatcher, protein ligation technology in antibody discovery. Please note the webinar is being recorded. Please do add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I'll now turn the show over to our speaker.
Thank you, Janice. Yeah, as you said, my name is Francisco Ylera, and I'm the R&D team leader at the Bio-Rad site in Munich in Germany. And in this webinar, I will present how many additional options you have by adding a tag to your antibodies, especially or namely the SpyTag. I will start with a slide about who we are, where we come from, introduce you to the SpyTag technology. Then I will describe our modular antibody assembly platform, the Trailblazer platform, explain to you how we use the SpyTag for an improved phage display selection technology, how we modified the SpyCatcher for the SpyLog technology, and how we apply it to the rapid generation of bispecific antibodies. And then in the end, introduce you to our Pioneer library, which uses all these different technologies for the generation of therapeutic lead candidates. So basically, the site in Munich was founded about 20 years ago. So we are focused on the custom antibody generation for our customers by phage display. And since then, we have developed more than 60,000 different antibodies. So basically, we have a very deep understanding of human antibody libraries and antibody selection methods. And for the last about 4 years, we also are a provider of SpyTag and SpyCatcher reagents. So what is a SpyTag technology? For all of you who are not familiar with it, the SpyTag is derived from a protein from Streptococcus pyogenes. That's where the name Spy comes from. And it consists of a small peptide, the so-called SpyTag, and a small protein, the SpyCatcher. And if you mix these two, then they spontaneously, autocatalytically form an isopeptide bond, as shown here on the right, between the lysine side chain and an aspartic acid side chain. So it's an isopeptide bond, a covalent bond. It is stable and it's irreversibly formed. And the great thing about this reaction is it is very fast and quantitative. Within seconds, you get your ligated product, and usually within 10 minutes or so, the reaction is complete. It's a very robust reaction, and it's also very specific. So if you do the reaction in lysates or within cells, it only takes place between these two partners and not with any other proteins. The SpyTag technology was developed by the Howard Lab at the University of Oxford, published first time in 2012, the version 1. In 2017, they published an optimized version, the SpyTag 2, SpyCatcher 2, which was optimized by phage display. And they claim that it's about 10 times faster than version 1, but it's still fully compatible. And here on the right, you can see a diagram which clearly shows that the reaction is faster and you get more ligation product with a version 2. In 2019, the Howard Lab published the version 3, which is again about 10 times faster than version 2, but maintains the compatibility to the previous versions. And here in red, you can see, again, it works much nicer, much faster than the previous version. However, the reaction is concentration dependent. So at low concentrations, as shown here in these diagrams, you see a very big effect. But if you switch to higher concentrations, as we usually do it for our antibodies, then the difference between these version 2 and 3 is actually quite minor. In this presentation, you will see data generated with SpyTag 2 and a mixture, sometimes SpyCatcher 2, sometimes it's SpyCatcher 3. But basically, the version 2 works quite well in our hands, so there's no immediate need to switch to version 3. So we developed a set of different SpyCatchers. A monovalent SpyCatcher, we've generated a dimer, the so-called BiCatcher, and we've used the SpyCatcher to the Fc domain of an antibody, the so-called ScCatcher. And we also generated versions with cysteines introduced, shown on the right, so that we can site-specifically label these catchers with melamide chemistry, so that we get a fixed degree of labeling and have a very high batch-to-batch consistency. And with our Fab antibodies, which carry a SpyTag at the C-terminus of the heavy chain and the set of different catchers and catcher conjugates, we can quickly generate all kinds of different antibody formats. So basically, we express a Fab once, and then we can couple it to the different catchers depending on our need for a respective assay. And the great thing of the reaction is it's the simplest protocol. It's just mixing the Fab and the SpyCatcher. It's a fast reaction. We usually say let it sit for 1 hour and it's done. And there are no additional steps required, no purification. It's fully scalable. You can do it just for an assay or at larger scale. And the great thing is, since it's a site-specific reaction, there's no modification of the antibody binding site. So usually we see an improved performance of these antibodies coupled to SpyCatcher conjugates compared to the classical conjugation of your antibody. So it has many advantages. You can take the best format for each and every assay. You can switch dyes for flow multiplexing assays. You can take all your antibodies and quickly convert them into the synthetic IgG format. Basically, within 1 hour, you can convert these into synthetic IgGs and continue with the testing. And then later, you can pick out the best candidates and convert them to real IgGs if necessary, or you can even stay in this format. So it is much more economical and it accelerates your workflow dramatically. So here's a slide with a Kumasi gel, which shows the SpyTag SpyCatcher reaction. So in the first lane is the Fab with the heavy chain on top with the SpyTag, and below the light chain. Next to it, the SpyCatcher. And then if we mix these two, then after 30 seconds already, we see the ligation product. And then as time moves on, we see that the product gets stronger, and at the same time, the heavy chain slowly disappears. And after 10 minutes, basically, the reaction is complete, and the heavy chain has shifted upwards to the ligated product. So it really works very nicely. So let's look at the assay performance of these SpyTagged coupled antibodies. And let's start with a direct ELISA. The antigen is trastuzumab coated to an ELISA plate. And then we detect it with an HRP conjugated IgG, conventionally conjugated to HRP. That's the blue curve here. If we use our BiCatcher HRP coupled antibody, it's the red curve, which is basically identical to the FCCatcher HRP coupled antibody. And you can see that these coupled antibodies are much more sensitive than the conventional IgG. So we believe that's because the picture here of the IgG with one HRP attached to it is not really representing the real world. With the random conjugation, you have some HRP sitting at the antibody binding site, then you basically have only a monovalent antibody left. And if both binding sites are conjugated, then you actually have a non-functional antibody. There are some antibodies which don't carry an HRP at all. There are some which only one HRP, giving only a weak signal. And the concentration of the fully functional, properly labeled IgGs is actually much lower than we think it is. And that leads to this shift of the blue curve to the right. So with our systems, it's all site-specifically labeled. Every antibody has the same number of HRPs, basically. So we have a better sensitivity, and of course, the labeling is also much faster. Here's an example for a Western blot showing that a polyclonal secondary antibody detection is not necessarily better. On the left is the Western blot with a bivalent Fab and detected with an gold anti-Fab HRP conjugated polyclonal antibody. On the right is our BiCatcher HRP construct. And there you can see at the strongest dilution, 3.1, you can still see a band. On the blot on the left, you cannot see that band. So it is not just faster because you don't have the secondary antibody incubation step and these washing steps. You also increase the sensitivity. Here's an example for the FCCatcher coupled antibodies. So we have different FCCatchers. We have human FCCatcher based on IgG1, 2, 3, 4. We have IgA, IgM, but we also have a mouse version, mouse IgG2A, and also a rabbit IgG FCCatcher. So that you basically can take your human Fab and quickly convert it into a mouse antibody, to be accurate, a human mouse chimeric antibody. And then you can use your standard detection reagents, anti-mouse Fc or anti-rabbit Fc secondary antibodies, which you probably have sitting in your fridge, and you can go ahead. In this example, I show that you can even use it for multiplexing. So I used 3 different antibodies, anti-cyclophilin A, anti-vimentin, and anti-Ki67, and coupled these to the human FCCatcher, the mouse FCCatcher, and the rabbit FCCatcher, respectively, mixed these 3 antibodies to stain UTOS cells, and then used species-specific secondary antibodies conjugated to different dyes. And as you can see here at the bottom, with immunofluorescence, we could nicely locate the 3 antigens. So it not only works nicely, it even works nicely for multiplexing. We also generated fluorescence catchers and used these to label or couple it to our Fabs, but as well to IgGs. So we can also add the SpyTag to IgGs at the end of the heavy chain C-terminus. And we compared it or evaluated it with dye light catchers. and compared it to our catalog products. So for that, we used an anti-CD3 antibody and stained PBMCs. And that you can see in these curves. So the green curve is the Fab coupled to the BioCatcher dialide conjugate. The yellow curve is the IgG coupled to the SpyCatcher dialide construct. And the blue curve is the monovalent Fab coupled to the monovalent SpyCatcher. So you can basically see for most, or for two examples of the three, the bivalent are slightly better than the monovalent, and the three coupled antibodies perform better than the conventionally labeled antibody which we currently have in the catalog. Of course, there are many more assay options. A few of them I want to show you. Typical task for antibodies is to generate sandwich assays. And if you generate antibodies, then you usually have all your antibodies in the same format. So you cannot use a secondary antibody for detection because it would also bind to the capture antibody. So that basically means you need to conjugate your detection antibody to HRP or some label. And this is so much faster and easier with a SpyTag technology. And as mentioned before, it's site-specific, so you don't have to worry that your antibody is lowered in performance because the binding site is modified. We can do screening after our antibody generation by phage display. We can do the screening in mono and bivalent antibody format in parallel, which is usually not possible. And the advantage of this is that the monovalent format, as you can see here, it shows a 384 well plate. And of course, you have less hits in the monovalent formats because you don't have avidity. So it is more stringent. So it's better to use a monovalent format to select the best candidates, the highest affinity candidates. But if you do the screening only in the monovalent format and you screen on some closely related antigens on which you do not want to see binding, you can do so. But if you then later switch to a bivalent format for your final assay, then it could happen that you suddenly see some cross-reactivity on the closely related antigen, which you see only in the bivalent format. So we can do the screening on the antigen in the monovalent format and screening on closely related antigens in the bivalent formats, and this way choose the best antibodies right away at the screening level. You can immobilize the SpyCatchers on different surfaces like Ylera plates, beads, resins, whatever, and then add your SpyTag antibody for an oriented immobilization, which reduces the amount of antibody which is needed and increases the sensitivity. And you can very rapidly and easily generate affinity columns by using SpyCatcher resin. You can simply run your SpyTag antibody over the column. It couples to the SpyTag and your affinity column is ready. And as I said, there are many more options. Yeah, just think about it, what you need and how you could use it. It's an interesting toolbox. With this, I want to switch to phage display and show you how we use the SpyTag in our so-called SpyDisplay. So phage display is a most established and most robust selection system for proteins, established more than 30 years ago. And it has several big advantages. One is that you can work with very large libraries with more than 10 to the 11 members, and it's a very fast protocol. In our case, we use it with a naive synthetic antibody library with defined germline genes, which are codon optimized. We have a large library compared to the natural antibody repertoire, which is about 10 to the 6 different antibodies per human. And we have a library of 10 to the 11. So basically that explains why we don't need the affinity maturation, which takes place in the immune system. The method is an in vitro method, so it is suitable for low immunogenic antigens, conserved molecules, immunosuppressants, toxins which would kill the mouse, vesicles, viruses, cells, protein complexes. It's quite flexible. Of course, we generate human antibodies because we have a human library. And the method briefly consists of immobilizing the antigen, adding the library, and then we go through several rounds of selection and amplification. And since it's an in vitro method, we have the option to add some closely related antigens to which we do not want to bind the antibodies to bind. We can just block the library by adding these proteins in excess, and all the antibodies which bind to these closely related antigens are then washed away. The specific antibodies are then eluted. E. coli is infected, and we can amplify the rescued antibodies sequences and prepare the phage for the next round. And after 3 rounds or so, we go to the screening step, screen for the specific antibodies, then we sequence those hits, and then comes the expression, purification, and QC, and the whole process takes about 8 weeks. We even have a fast track options, then we can do it in 4 weeks, the full process from starting of the selection to the purified antibodies. So in conventional phage display, you have your phage met shown here in the E. coli as a circle, which carries the Fab fused to the P3 phage coat protein, genetic fusion. So after infection of the E. coli with helper phage, which adds the DNA for all the other phage proteins, the E. coli produces new phages, which carry the Fab covalently on the surface of the phage coat. And inside the phage, the phage mit is packaged. So we have this linkage of phenotype and genotype, which is required for the selection. In SpyDisplay, we also have a phage mit, but it carries only the Fab, no other elements needed for phage display. We use a special strain, which we modified, which carries the SpyCatcher P3 fusion protein in its genome. And again, we need the helper phage infection to add the other phage proteins. And then the E. coli produces new phages, which carry a SpyCatcher on the surface of the phage. And from the phage mit, we get our Fab with a SpyTag. And then within the E. coli, the SpyTag and the SpyCatcher ligate. And this way, we get our Fab covalently displayed on the surface of the phage. And this method has several advantages. First of all, the library is already in the expression vector, so there's no subcloning step required between the selection and the screening step. Less steps means it's less error prone, it's faster, but we also further improved or accelerated the selection protocol so that we can now do 1 panning round per day. The Fabs are obviously expressed without the fusion of the P3, so it's a much smaller protein. And as you know, the expression of mammalian proteins in E. coli, the folding is not the easiest for the E. coli protein folding machinery. So we get more properly folded Fabs onto the surface of the phage. Obviously, the resulting antibodies all carry a SpyTag, so they are compatible with our Trailblazer platform, the Modular antibody assembly platform, which I showed. And we showed that we have a truly monovalent display for the selection of high affinity antibodies. That means that every phage carries a maximum of one Fab. But we can also switch to a polyvalent display where every phage carries in average 2 to 3 Fabs. And make use of the avidity for the selection of antibodies against difficult targets. There are some additional advantages which are not needed for the antibody selection. You can use this SpyDisplay for the selection for fusion of the protein of interest at the N-terminus to the phage code protein. With the conventional method, you always have to fuse it at the C-terminus of your protein. But since you can place a SpyTag at the N-terminus or the C-terminus or even within the protein, you have more options with SpyDisplay. And you can also use the TUT translocation pathway for your phage display. And so the SAC pathway is what we use for normal phage display and for our antibodies. It transports the unfolded protein to the periplasm. And this is required for the P3. For SpyDisplay, we can, the P3 and the protein of interest is transported independently. So we can use the SAC pathway to transport the P3 into the periplasm and use the TAT pathway, which transports the folded protein to the periplasm for the protein of interest. And then they ligate in the periplasm and then get displayed on the surface of the phage. And we showed this for GFP, which only efficiently matures in the cytoplasm and expresses its fluorescence. So if we use FOA, a signal sequence for the SAC pathway, then we get only very weak fluorescence as shown by the epi on the left. The tube in the middle shows the TAT pathway, bright fluorescence, and on the right is the negative control. Here's a slide which shows an affinity maturation selection. And with this slide, I basically want to show or emphasize how well the SpyDisplay selection platform works. So in the last over 30 years, a lot of modifications and improvements of phage display have been published. But there are also quite some which have been published and then never seen again. Because they basically do not really work so well. So here I chose the affinity maturation because if I would show it with a Pioneer library, you could say the Pioneer library is so great and that's why it works so well. So I used affinity maturation, which usually has standard sized libraries of 10 to the 8, 10 to the 9 different members. So it's more comparable to other maturation campaigns, I would say. So in this example, we started with an antibody with an affinity of about 1 nanomolar, and the second example, an antibody with about 600 picomolar affinity. So quite a good affinity already. We exchanged the heavy chain CDR2 and light chain CDR3 separately, so 2 libraries for each antibody. And then did the phage display selection. And what we got out of this are shown here with these green dots. In red is the parental antibodies. So you can see that the affinities plotted on the y-axis is better for all the antibodies which we analyzed. And there are a lot of antibodies, quite a good number of antibodies with affinities down here at the 1 picomolar level, shown here by this sensorgram measured by BLI. Basically, by BLI, we cannot measure this affinity anymore because the off rate is so slowly that yeah, we said here it's a affinity of below 1 picomolar. And this for both examples. So it basically shows that with SpyDisplay, it works very efficiently and you can select ultra high affinity antibodies. Okay, with that, I want to switch to SpyLog, which we apply to the rapid generation of bispecific antibodies. So SpyLog is a reversibly inhibitable SpyCatcher, which we developed. We introduced a mutation, a serine to cysteine mutation at position 59. And we showed that the SpyCatcher, which we call SpyLog after this mutation, is still fully catalytically active. So if we add a Spytagged fab to SpyLog, then we get the coupled product. However, if we let this cysteine of this SpyLog react with a small disulfide forming reagent like Ellmann's reagent or with this activated biotin, then the SpyCatcher is inhibited and does not react with the SpyTag anymore, shown here or symbolized here with this lock around the SpyCatcher. And by adding a reducing agent, we can cleave this disulfide bond and our SpyCatcher is active again, shown here by the formation of the coupled antibody. So the chemistry is pretty straightforward. We have the cysteine, the thiol group in the cysteine. We let it react with DTNB, Ellmann's reagent. We get the disulfide bond, which locks the SpyCatcher or the SpyLog. And after reduction and cleavage of the disulfide bond, it's fully functional again. And that's shown here on this gel. In the first lane is MBP with a SpyTag. Next to it is our SpyLog. And if we mix these two, it's a closed SpyLog. And if we mix these two without reducing agent, then we don't see an ligation product. Only after overnight incubation, after 16 hours, you can see a weak band. So it shows the reaction is not completely inhibited. It's just slowed down dramatically so that we can nicely use it to lock the SpyCatcher. And if we add a reducing agent, in this case TSAP, then you can see that after 5 minutes or so, we start seeing a ligation product. And after 1 hour, 1 to 3 hours, the reaction is usually complete. And how do we use it for bispecific antibodies? We generated a fusion protein of the normal SpyCatcher and our SpyLog. If we then add a first antibody, it can react only with a standard wild-type SpyCatcher. We can then add reducing agent to unlock the SpyLog, add the second antibody, and it then couples to the SpyLog, and we get our bispecific antibody. Basically, we have 2 methods, a quick method and a purification method. In the quick method, we get the product in about 90 minutes. With the purification step, if we use biotin to lock the SpyLog, we can purify the first coupling product on streptactin columns, remove any excess of Fab1, then add a reducing agent, release our first coupling product, unlock the SpyLog at the same time, add the second Fab, couple it to the bispecific antibody, and then we can purify the excess of Fab2 with a SpyCatcher column. So it works very nicely. We get very pure bispecific antibodies, and yeah, it can be done in high throughput. And we have applied this for the generation of PD-1, PD-L1 antibodies. We chose 4 anti-PD-1 antibodies and 9 anti-PD-L1 antibodies. So we formed 36 different bispecific antibodies and tested these in an functional cell assay. So basically, PD-1, PD-L1 interact with each other, and PD-L1 basically inhibits the immune system. So it's an important target for cancer immunotherapy. And if the interaction is inhibited, then we see yeah, an improved immune system, or in this assay, we see that luciferase is expressed by the PD-1 overexpressing cells, and that gives here the signal. We then on the left for the 36, and then we on the right we looked at it for one bispecific antibody. So here the purple curve is a negative control, GFP monospecific antibody generated with this SpyCatcher SpyLog tool. In blue is our PD-1 Fab, so it gives only a very weak signal. In red is a PD-L1 antibody. In green, the mixture of the PD-1 and PD-L1 antibody. And in black is a bispecific antibody. And you can see very clearly that it is gives a much stronger luminescence signal. Okay, and now I want to introduce you to our new Pioneer library. It's a new state-of-the-art phage display library. It's one of the largest functional phage display antibody libraries ever made. We optimized it for phage display so that ideally all antibodies can be selected by the method, whereas for most other libraries, you observe a large portion which is basically not selectable by phage display. So it's a dead volume. We optimized the library for the generation of therapeutic lead candidates. So we chose selected framework genes. We have a CDR design which is close to nature, and we optimized the library for good antibody developability so that we get antibodies out of the library which perform well as a lead candidate and do not need to go through many rounds of optimization. We use our SpyDisplay selection method, which I explained to you. The resulting antibodies carry a SpyTag, so they are compatible with the Trailblazer antibody assembly platform for faster and more versatile screening of the resulting antibodies. And with the just shown SpyLog technology, we can very rapidly generate bispecific antibodies for screening. So how does this library perform? Here's a test selection on 4 antigens, and for about 20% of the antibodies which we analyzed, the affinity is in the sub-nanomolar range. So we get really excellent affinities right out of the Pioneer library. And we then generated lead candidates. We chose TIGIT, another immune receptor, which plays a role in cancer immunotherapy with over 30 clinical trials ongoing for our example, and generated antibodies, and then compared these to antibodies which are currently in the clinic, in the phase 3. And we could show that we have a great diversity. We got antibodies against 8 bins for a protein which is quite small. The extracellular domain has only 16 kilodaltons. And we got antibodies with excellent properties and developability directly out of the library without further optimization, which is shown here for one example. Of course, we have many more. So we have good affinities, good TM values. All antibodies were above 73 degrees. Most antibodies had a good monodispersity, a low self-interaction propensity. All tested antibodies but one had a low hydrophobicity, passed the polyreactivity screening, were stable for 4 weeks at 37 degrees in the stability test. We tested these in a functional cellular assay and had similar properties as the antibodies in the clinic, and had a very high germline homology. Actually, all our antibodies were better than vibostolimab, which is a phase 3 candidate. So, which basically shows that we have excellent properties of our antibodies right out of the library, and we expect that it helps saving a lot of time, which is currently needed for these optimization rounds. And that brings me to the summary. So I showed you the Trailblazer platform, our modular antibody assembly. We can very rapidly change the antibody valency, the species or the isotype of the antibodies. It's great for rapid site-specific labeling with our prefabricated SpyCatcher conjugates. And it gives rapid access to many more assay setups. With a SpyDisplay selection technology, we have a very fast and efficient selection technology, which is very well suited for the selection of high affinity antibodies and offers some additional new options like the N-terminal display or TOT display using the TART pathway. And with the Pioneer Antibody Discovery Platform, we have one of the largest functional antibody phage display libraries generated, if not the largest. We have a high hit rate. We have a great antibody diversity after pannings, and we get excellent affinities right out of the library. We have a very good antibody developability and fast selection and screening through the Trailblazer technology. And with the SpyLog technology, we can very rapidly generate bispecific antibodies with high purity and yield for screening of the best antibody pairs. And here it's important to keep in mind, if you have 10 antibodies 1 and 10 antibodies 2, and you want to generate these pairs, then you already have 100 bispecific antibodies to look at. So it's a large number which is feasible for the SpyLog technology, but not so feasible for most other bispecific antibody production platforms. So how is it all available to you? So we offer these SPY, BY, and FC catchers for our custom antibody generation projects. But we also offer these catchers in our catalog. The biotin and the HRP conjugates are available already now in the catalog. The dilite conjugates which I showed are not available in the catalog yet. They are available for the custom project. In the catalogs, they will probably follow next year. And we plan to generate many more conjugates which will be launched in the future. So look at our catalog. They are not expensive. That's a really convenient tool if you work with SpyTag. It's available for research use only. So if you work in a company, in a commercial setting, then please contact us for licensing options. And I'm sure we find an interesting way for you to work with it. And with this, I want to say thank you for your interest, and I'm happy to answer your questions.
Excellent. Thank you so much. We do have a few questions. First question. Nice talk. Is the SpyCatcher antibody construct expressed cytoplasmically or paraplasmically?
The SpyCatchers are expressed cytoplasmic. In E. coli for the standard SpyCatchers, if we look at the FC catcher, then we go to mammalian expression because the FC part is glycosylated. So we want to have the FC part as it is in IgGs.
Next question. Any reason why the cysteines were introduced to SpyCatcher? Is it possible to completely remove or replace the cysteines and still retain binding strength?
The SpyCatcher is a protein which does not contain any cysteines normally, naturally. So we don't need these cysteines for its function to ligate to the SpyTag. We introduced these cysteines for site-specific conjugation. So all our SpyCatchers carry the cysteine at the same position, are labeled through the cysteine, so have the label always sitting at the same position, and we know what is the degree of labeling of the catcher.
Next question. Is there a particular trick to getting high affinity antibodies, which you were talking about when you were discussing the phage display?
Yeah, so to get high affinity antibodies by phage display, there are basically several aspects which play a role. So first, it's a selection method. You want to have a selection method which works very efficiently, which has not many biases, like different growth of the E. coli during the amplification step. You want that your antibodies are equally well expressed and displayed on the library. You want to get rid of empty phages which don't carry an antibody, which can be carried from one round to the next, and basically make the selection step less efficient. So that's what I wanted to show with SpyDisplay, that it works very efficiently and is great for the selection of high affinity candidates. But there's more than the selection technology, of course. The second important step is that you have a high quality library, so that ideally the whole library space is really filled with sequences which can be displayed on the phage. So you always have some cloning artifacts, stop codons, and so on. But it's important to keep this fraction as small as possible, and to also work with sequences which behave well in E. coli. So can be expressed, can be folded, and end up onto the phage. Otherwise, you might have a large library, but half of it is actually never displayed on the phage. And then, of course, the library size is very important. So the larger the library, the more likely it is to get high affinity antibodies. And as I said, Pioneer is the largest or one of the largest phage display libraries. So that is, in combination with the other factors, the trick why we get such great affinity antibodies right out of the selections.
Perfect segue into one of the questions. One of the questions is, how did you determine the size of the library?
So here it's important to keep in mind that we work with a synthetic library. So the CDR cassettes were synthesized by trinuclear mutagenesis. So the design of the heavy chain CDR3, for example, has a theoretical diversity of more than 10 to the 25. So of course, we cannot synthesize that many heavy chain CDR3s. So what we basically get out of synthesis is about 10 to the 17. So it's a huge number. And if we clone these 10 to the 17 cassettes into our library to get 10 to the 11 colonies, then it's just extremely unlikely to get one antibody twice. It's like winning in the lottery, literally. That basically explains, and of course we have diversity in the other CDRs as well, which increases or decreases the chance of finding an antibody twice. So basically the number of transfectants which we got is equal to the library size. And of course we also confirmed this by NGS sequencing. We sequenced about 12 million clones, and basically we couldn't find any duplicates in the library.
One more question about the phage display topic. How does the elution of the SpyCatcher work during phage display?
So we introduced a protease cleavage site between the code protein and the SpyCatcher. So after washing and removing all the unspecific antibodies, we simply cleave off the phage from the fab and elute it, and we can continue with the E. coli infection. So it's independent of the antibody affinity, so we can elute even very high affinity antibodies.
And our last question is on the topic of the bispecific antibodies that you discussed. Can the bispecifics made with SpyLock be used as drugs?
The SpyCatcher is a bacterial protein from Streptococcus pyogenes. So you basically don't want to give a bacterial protein into humans. It is done actually for vaccines, but there you want to get an immune response for your drug. You don't want to get it. So it's not really meant as a bispecific as a drug. But as I described, there is a very large number of candidates which you can generate very easily, very rapidly with the SpyLock technology for a first screening step to find out which candidates actually show the activity you are interested in. So for 10 antibodies A and 10 antibodies B, you can screen these 100 bispecifics and then maybe produce the 10 best candidates in your final format and move on with that.
Good approach. Very good approach. With that, that was our last question. Oh, wait, we have one more. How are the advantages of the SpyCatcher technology in comparison to SpyDisplay?
Yeah, SpyDisplay is what we use for the UCAR libraries, for our research and diagnostic projects, library we used for decades. Maybe for those who are not familiar with it, in SpyDisplay, you have a free cysteine on the surface of the phage, code protein P3, and you have a free cysteine at the C-terminus of your fab, and then they form a disulfide bond and also lead to a covalent display of the fab on the surface of the phage. And you can easily cleave it with a reducing agent. So it's a great technology, but it also has some minus points. So one thing is in the SpyDisplay, the selection system and the protein of interest, the library, are not separated on the phage mid. So you need to subclone the library after selection into an expression vector. It is slower. And you not only get the disulfide bond formed between the fab and the P3, you also have P3 dimers, You have Fab dimers, so it is not that easily controllable. The display weight is lower than in SpyDisplay. I mentioned a monovalent display, which basically means you have more empty phages with this display than with SpyDisplay, and empty phages basically can stick to your antigen and are unwanted. Yeah, there are some more topics, but these are the main ones.
All right, well, thank you very much. I think we do not have any more questions. So with that, in concluding, I'd like to thank Dr. Ylera for providing these insights into the use of SpyTag as well as SpyCatcher, protein ligation technology that is used in antibody discovery. And I thank the attendees for joining the webinar today. An on-demand version will be available within a day or so. I'll send a link to it via email to everyone who registered. Please do feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.